Hello everyone, my name is Xubo Tang and I'm come from the City University of Hong Kong and my supervisor is Dr. Yan Sun. I'm very glad to be here to share our work. And the topic of our work is identifying plasmid contigs from metagenomic data using transformers. So what is a plasmid? The plasmid is the small and circular piece of DNA mainly found in the bacteria. And plasmid is also a carrier for some accessory genes, such as the antimicrobial resistance genes or some metabolic genes. And the plasmid can be transferred from one bacteria to other bacteria, and it plays a key role in the bacterial evolution. It is very important to study the plasmid because the plasmid are carriers for horizontal transfer between the bacterial genomes, and the plasmid can carry the receptor genes, including the AMR gene. And we know if the bacteria contains the AM gene, it will make the drug ineffective. And here is the diagram showing the race of the resistant bacteria. And we can see from the 1985 to 2013, there are many resistant bacteria. So studying the plasmid can shed the light on understanding how bacteria obtain these AMR genes and how they transfer between different bacteria. To isolate the plasmid, the first way is using the whole genome sequencing. We can grow the bacteria in the lab, and after assemble the contig, we can identify the plasmid easily and accurately. But very few of the plasmid can be grown in the lab. And another more efficient way is to using the metagenomic sequencing. We can collect the sample from the patients and environment, and after we assemble the contig, we can identify the plasmid with high diversity more efficiently. Using this method, we can also identify the plasmid which is novel to our reference database. So our problem formulation is to identify plasmid contigs assembled from the metagenomic data. So after we obtain the metagenomic sequencing rate, we will assemble the rate into contigs. And we use this contig as input, and we can identify which contigs are from the plasmid. There are two main challenges. One challenge is that the plasmids are very diverse. Here is the histogram showing the pairwise similarity of plasmids, and the x-axis is the similarity. Y-axis is the count of the pairwise alignment scaled by the log. And the left bar is the number of the pairwise alignment, which is 0. It means the majority of the plasmid pairs share little or no similarity. And another challenge is due to the gene transfer, plasmids share the genes with the chromosomes, which make it more difficult to distinguish the plasmid from the chromosomes. Nowadays, there are 3 types of methods to identify the plasmid. The first method is using the alignment-based method. They align the query contigs to the reference database using the stringent threshold. And we found these tools have high precision but low sensitivity. And another method is using the learning-based method. They will count, for example, the chimer frequency, and using this chimer frequency as a feature vector. Using this chimer frequency feature vector to train their machine learning models to distinguish the plasmid and chromosomes. And we also found that this type of method has low precision but have high sensitivity. And there are also some method we call hybrid method combine the alignment-based method and the machine learning method. We will use the alignment to obtain the feature of this theory contigs. For example, the number of hit genes and the hit marker or hit protein, and encode all these alignment into feature vectors, and using these feature vectors to train the learning models to classify the plasmid and chromosomes. And we found this type of method usually achieves optimal trade-off between the sensitivity and precision. Here is the pipeline of our tools. And the input of our tools is the assembled contigs from the metagenomic data, and the output is the putative plasmid contigs. To build the database, we download the plasmid from PRCD and build the plasmid database. And also, we predict the proteins using Polygon to obtain the plasmid proteins. And we cluster all these plasmid proteins into protein cluster and build the plasmid protein cluster database. So after we encode the assembled contigs, we will first align the contigs with our plasmid database with stringent thresholds. For example, we can see both the coverage and identity as 90%. Now here we can identify the plasmid contigs share high similarity with our reference plasmid. For other contigs didn't satisfy this threshold, we will align the protein of this contigs to our protein cluster database. And we encode the index of the aligned protein cluster into the feature vectors. So here, actually, we treat each protein cluster as a token, and we encode this feature vector into our transformer model. Because different orders share low similarities, so we build the models for each order. So finally, actually, we encode the encoded feature vector as a sentence into our order-specific transformer models to predict the putative plasmid contigs. Besides using the protein cluster as tokens, we can also use the nucleotide or amino acid as tokens. For example, after we obtain the contig, we can use the nucleotide base pair encoding to encode this contig into the sentences. The nucleotide base pair encoding actually is to calculate the high frequent combination of nucleotides. So we can use these primers with different lengths as a token to encode the contig into a feature vector. And also we can translate the protein from the contig. For example, we can use the amino acid as tokens. So we treat different amino acid as different tokens to encode the proteins into the system. And also, we can use a similar way with the nucleotide base pair encoding or the amino acid base pair encoding to obtain the high frequent combination of amino acids as a token to convert the protein to the feature vector. But if we use this type of tokens, we still need to use the majority vote. For example, if we use the nucleotide base pair encoding, because the contigs may be very long, so we need to use the window to cut this original contigs into several pieces and predict the origin of different pieces, and then use the majority vote to say if the original contig is from the plasmid or not. Also, for the proteins, because one contig may contain multiple proteins, so we need to predict the origin of the proteins and use the majority vote to say if the contig is from the plasmid or not. Here we compare the performance of different tokens including the protein cluster, amino acid base pair encoding, and also the nucleotide beta encoding. And we can find only the protein cluster token can capture the importance of proteins and genes. And also, the protein cluster token is the only method can capture the correlations between the proteins. So we can find in this experiment, the protein cluster tokens can achieve the highest precision, recall, and F-score. We compared our method with other tools on the simulated dataset. Here we use the Chemi2 marine dataset, and this dataset contains short-read simulated metagenomic data. And we can find our tools, which has been highlighted called plasmid, achieved highest F-score. And we can see for these tools, for the alignment tools, for example, the Morica and Platon, because they use the stringent alignment threshold, so their recall, the yellow bar, is not high enough. And for this machine learning-based method, for example, the PPR method or PlusFlow, because the data set contains many chromosomes and their precision is not high enough. So in the Chemi2 data set, their precision is not high. Here, we interpret the performance of transformer by analyzing the attention distribution of different tokens. Here we visualize the tokens in one sequence. And in this matrix, the light color means this token has high attention with other tokens. And in this matrix, we can see the protein cluster 16138, it has high attention with other tokens. And here we count the top 50 important tokens, and we use the interpro to annotate the function of these protein clusters. And we can see like the function of over half protein clusters are unknown. And for the protein cluster with known functions, many of them are related to the protein class, the plasmid core function. For example, 5 tokens are related to the conjugation, and there are also the protein cluster related to the replication. So here is our conclusion. So our method combines the alignment-based and learning-based method and achieve the optimal trade-off between the sensitivity and precision. Also, we use the protein cluster as a token in a transformer. And using the protein cluster as token, we can capture the importance of different proteins and their correlations. And also, based on the interpretation, we can find the protein which plays an essential role in the plasmid also are more important for the classification. Thanks for your attention.